cancer, AIDS, diabetes, hypertension, infectious diseases caused by different pathogens. These are all major health hazards. Many diseases still remain uncured because of their resistance towards the existing drugs and very high antigenic variability. This necessitates discovery of a new drug. In the ancient time, people used very simple things. Any source of information right from folklores was used for the drugs. The plant products was the first natural choice. For example, drugs like morphine, reserpines were all plant products. Natural selection during evolution and competition amongst various organisms created many products, natural products, for example, molds, which created some coat against them, which created resistance to external other infections and bacteria. Many drugs were developed from these natural products. Examples are quinine, penicillin, and so on. Another approach in the older time was to see the comparison of the activity of whatever chemicals were available. Drugs like thinomycin were developed by that method rather. When number of drugs belonging to the same series were available, then people tried to check their activity against a disease and tried to find out a compound which will yield maximum activity. Sulfur drugs, for example, were developed by that method. Then came chemical era. In the chemical era, people used many physicochemical methods like comparing chemical structure with the activity and so on, and drugs like cephalosporins were developed. Biochemistry was very much useful. People started understanding mechanism of drug action and they tried to design or find out, discover new drugs based on the mechanisms. A typical example can be given of carbonic anhydrase inhibitor and suicide inhibitor. Thus, there was ancient era where the plant products and natural products were used. Then came chemical era where chemical structure activity relationship were used. Then people started become more rationalizing their drug discovery process, and then came the biochemical era, and ultimately we go to the rational approach to the drug designing. But in these high throughput methods, combinatorial possibilities are very high. Just for the case, we take a simple example, a benzene molecule, a disubstituted benzene molecule with the 4 open positions. Suppose we want to try 10 different substituents on these molecules at all these 4 places, then we have to synthesize 10,000 compounds and test all of them. What was the result? The result was the cost of bringing a new drug into the market became very high, from 500 to 700 million dollars. Again, there was very big competition among the drug companies. The time taken was also very high, 10 to 12 years. The manpower needed was also high, 200 to 300 people. Bioinformatics, computer aided drug design can reduce not only cost, but manpower and time needed for a particular drug discovery. Let me give you a simple example. Example tried at Seneca in Italy. It is known as the Dructin project, where they tried to discover the peptidomimetics active against cell growth and migration, and they brought down the cost from 270 million euros to 150 million euros. Now, this is very, very important for India, because we have a very big pharma market, which is about 350 million US dollars and so on. So, we should go for rational and computer aided and systematic drug designing. What was the first rational approach to the drug designing? There was a method known as QSAR, that is quantitative structure and activity relationship, very popular amongst all the pharmacologists in the older time. In 1970s, people took activity, something as minimum blocking concentration, IC50 values or cell survival or so on, and then compared it with simple parameters like molecular weight, boiling point, melting point, partition coefficient, and developed a mathematical relationship between the activity and these parameters, and the method was very successful. They really improved the designing of a new drug in a dramatic way. However, there were limitations, because these parameters were not directly correlated with the activity of the drug, because we did not know how the drug activity was, what was the reason, physicochemical reason, but randomly chosen parameter. So, people started becoming more systematic, and second generation QSR came into practice. People tried to use parameters which were more systematic, obtained from quantum chemical calculations. There was quantum chemistry program exchange in Indiana, USA, which freely supplied these programs, and people calculated using these methods: highest occupied molecular orbital HOMO, lowest empty molecular orbital LEMO, total charge, super delocalizability, bond order, and so on. And these parameters were later compared with the activity, and these parameters were known as descriptors, and many descriptors were getting discovered and used on. This really gave birth to the very recent method, which is known as 3D QSR, where the descriptors are calculated not from quantum chemical calculations alone, but using 3-dimensional structure and topology based parameters, and the method is very, very successful. What is really then a rational or a structure based drug design? The rational is a bad word, it does not mean all other methods are irrational. This is basically structure based drug design. There is a simple assumption that for every drug to act, there has to be a target molecule, and the drug molecule binds to the target, and the activity is given. One which gives the maximum activity is the most efficient drug. The target can be anything. Majority of the targets chosen were the proteinous molecule. They could be enzymes, receptors, circulating messengers, storage proteins, molecules involved in synthesis of DNA and protein synthesis, and so on. The DNA molecule, RNA molecules were also very good targets. There were many drugs which bound to the DNA molecule, stopped its replication, and these were very popular in the cancer treatment. There was a time when cancer drug was synonymous to a DNA intercalator drug, and there are very different kinds of molecules which are designed as a DNA binding drug rather. Another big class of drug was membrane bound molecules and membrane transmembrane channels. For example, sodium channel and inhibitors of sodium channels are all anesthetics, the poisons, the toxic molecules, whereas inhibitors of calcium channels and activators of calcium channels, these are involved in controlling of the blood pressure and hypertension. There are many other molecules sitting at the membrane site, and they are very good targets for drug designing. What is really the objective in this drug designing. It is to discover a molecule which is suitable to bind to this protein molecule and can give you maximum activity. So, what are the steps involved in this? The first step is we must get a therapeutic target, get its structure. Then we should find out at least one compound, this compound is known as lead molecule, which shows some little activity against this therapeutic target. Then we try to optimize by this ligand molecule or lead molecule by any method and get a molecule which will show the maximum activity, and this is how the drug candidate is developed. There are many other steps into it, because first you have to go to chemical assay methods, then you go to animal test, then there are 3 stages on clinical trials, the first level terminal patient, healthy patient, then the population which is free volunteers, and so on. Now, we come to the physico-chemical principle in drug design. This was known again from long time. It was embedded in Emil Fischer's lock and key hypothesis. What was it? That every drug molecule is like a key, and the target has an active cavity which is like a keyhole, and the key must fit into this keyhole properly to give the maximum activity. It was very simple, and not only that, it is the most essential condition. It is called steric compatibility between the active site and the drug molecule is the first necessary condition for a drug design. But it is not a sufficient condition, because the drug molecule acts with the receptor active cavity not only by steric force only, but there are electrostatic interactions amongst the positive charges and the negative charges, hydrogen bonds between the electron donor and the acceptor. If there are ions, then ion mediated interactions are there. If there are water molecules, then water mediated interactions are there. The target molecules are also not as rigid as shown in the Emil Fischer's lock and key hypothesis. These are made up of thousands of atoms connected to each other by bonds, governed by chemical rules of bond length, bond angle, torsional angle variability, and so on. At a room temperature, the atoms are moving and they are in the dynamic state of equilibrium, and all these things should be taken into consideration while computing a parameter for interaction of the ligand with the receptor or a target. The ligands were also not simple. They also had several tautomers, series of conformations, that also should be taken into consideration before computing the chemistry based or the structure based parameters. How to obtain the target structure? That was the first hurdle in this kind of approach rather, but now things are changed. In the last 2 decades, X-ray crystallography, nuclear magnetic resonance, also theoretical modeling coming in a big way, have created structural data on more than 33,000 structures. These are available at the protein data bank at rcsb.org. One of the big problem is target validation, because many times we feel it is a target, but target has to be validated, otherwise the whole designing goes wrong. Another big problem was the assaying, that IC50 minimum blocking concentration were not good parameters. The chemist had better parameters based on chemical kinetics, like Michaelis-Menten equation gives relationship between the substrate concentration and the rate of reaction, and one can calculate association constant, dissociation constant, and use them into the activity as a parameter. Even biological assay methods have also substantially improved. Large scale assay methods are available because you want to screen thousands of natural products and so on. What are the methods of drug designing? For that we have to see what are the possibilities. Generally there are 4 kinds of possibilities. In the first case, you may not have any structural information either on ligand or a drug or on a target molecule. In such a situation, there is no other alternative than using classical old QSR method. The only modifications one can do, use diverse subsets, say that is the use chemicals belonging to different classes, and use more refined statistical methods than the old ones. Suppose we have knowledge about the 3-dimensional structure about the drug molecules and or the ligands or the lead molecules, but no information on the target or its structure. In this case, and which is the most popular case, very often you end up with this case, 90% designing is done by these methods. We go to whatever is called nowadays 3D, 3-dimensional QSR. Here, based on the 3-dimensional structure of the molecule, we develop several parameters and these are fed into QSR equation and solved. There is also method called pharmacophore search or structure based pharmacophore search and so on. Third situation is we have information on target, we know target structure, but we have no information on drugs or what lead molecule is. In that case, there are 2 alternatives. In the first case, we can do is search all chemical database library, and chemoinformatics has a big role to play here. In the second case, we can go inside the cavity and try to design molecules, this is called de novo design, and get a new drug molecule. There is only a joke that never use de novo methods for de novo drug design. The reason is very straightforward and simple, that you may design a molecule, but it will never become a drug because of some other consideration. So, these methods are usually used to get the basic scaffold, and that scaffold is screened against the chemical libraries, and then it is consideration of the pharmacologist and other people, we come out with the drug candidate, and which is a very successful approach. Lastly, suppose we know both the structure of the protein on target and the ligand structure. In that case, we should calculate free energy of interaction between the protein or the target and the ligand, and one should compare free energies of interaction and compare with the chemical assays which we get rather. Only problem is this is a very computationally intensive task. Just for optimizing one ligand would take a week's time even with the fastest computer, and which is a major bottleneck, although the method is the perfect method. The pharmacophore search method I was telling, that we take any kind of diverse compounds, we show activity against some target, find out their 3-dimensional structure. There are good databases like Cambridge crystallographic database and so on. There are now several databases on structures of small molecules, and people are creating computer model based design structures database and use them. You calculate some chemical parameters and overlap these structures on one another. Then you try to find out some essential common features like presence of aromatic ring, charge positive centers, charge negative centers. Then use this information as a template to search for the database and then get the drug candidate. The target structure based drug design which we were discussing is a very complicated method. The first is we should optimize the protein structure shown here. The next is we should look for the active cavity and then try to think about what are the important features of this cavity like aromatic rings, donors, hydrogen bonds or some electropositive, electronegative, and then try to make a mental picture or make a mental template also, and then search for the compounds which will fit it. The compound is moved in different direction, different conformations of the compounds are tried, and ultimately the best compound is selected. A typical example is dihydrofolate reductase inhibitors designed by a combination of crystallography and designing methods rather. What is a pharmacogenomics, which is one of the most important field of the present time? We take a gene sequence which shows some importance related to a particular disease, translate this sequence into amino acid sequence, make a protein, find out its structure if available in the database. If not available in the database, then we model the protein and then find out a ligand which will show the activity against this or maximum binding. We calculate its free energy, compare its free energy of interaction and find out. This rational drug designing which I have discussed so far would allow us to enjoy the fruits of human genome project, and this is our ultimate objective. And this project was one of the most expensive project of the last decade, and the objective was we should be able to give better service to the mankind, and pharmacogenomics is going to solve this problem. Generally, we have four kinds of situations. The first situation is we do not know either the structure of the target molecule or the structure of the ligand molecule. In this case, there is no other alternative than to use the old QSR method, go on screening number of compounds and try to optimize the activity. Suppose we know three-dimensional crystal structure of the ligand molecules, but we have no other information on the target molecule, and this is usually a case that we know that many ligands they act for a particular disease, but we do not know their mechanism, we do not know their targets, and we do not have their three-dimensional structures. In this case, what we can do is that one can use two-dimensional and three-dimensional QSR technique. 3D QSR is based on generating topologically based descriptors. The QSR equation is set using all these topological descriptors, and very sophisticated techniques are used for analyzing them, and one can optimize the activity and design a new ligand. This is still one of the most popular method used by modern pharmacologists and drug companies, and it is highly based on the chemoinformatics databases available. If we are lucky enough, we know what is the target molecule, and we also know its three-dimensional structure, but we do not know which ligand will bind to it better. In this case, there are two options. First, we can go to chemoinformatics databases, select few compounds and do docking method, which I will discuss later, and try to calculate interaction and some say parameters, and based on that we can design a new molecule. Another method is de novo design, that is, we do not care about the structural database for the ligands, but we try to build a molecule using laws of physics chemistry inside the active cavity of the molecule. For that we first understand the nature of active cavity and then try to make molecule, and peptidomimetics is one of the most popularly designed inside the active cavities. However, there is a joke where people say that don't use de novo method for de novo design. It is not joke, the reason is quite obvious, that if we just design a molecule inside the computer, it is quite likely that that molecule either may not be synthesized, or that molecule may not show any pharmacological activity if synthesized, because it may have a different structure. So, what we do is usually like peptidomimetics, we design a molecule inside the active cavity and use it as a scaffold, and then to search the chemical databases, the chemoinformatic databases, and then look for the activity or some other pharmacological considerations, and then design this molecule. In case we are lucky enough, we have knowledge about the 3-dimensional structure of the target molecule. We have also the knowledge about the 3-dimensional structure of the ligand molecule and its conformational flexibility. We can go to calculation of free energy. Thus, all the drug designing methods fall into two classes, if we leave aside the just QSR, old QSR. First is called analog based drug designing method. This does not depend upon the knowledge of the target molecule or its structure, and it is basically optimize a lead molecule by consideration of its physicochemical properties and topology. One of the most popular method in this class is pharmacophore search. What we do is, we take a target molecule, we take a ligand molecule, we take different ligands which act on a particular disease, chemically diverse molecule, see their 3-dimensional structures, calculate some of their physicochemical parameters, set charges, etc. Then try to overlap these molecules on one another, and once we overlap, although the molecules might belong to totally different chemical series, some principle evolves, like presence of aromatic rings at some places, presence of some positively charged groups at some places, presence of some negatively charged groups of some places, and so on. This is a old pharmacophore method put into the new 3-dimensional approach. Now, we can use this as a template and then try to search the structural database. In other case, we can go in a more elaborate 3D pharmacophore search. In target structure based drug design, we try to do docking, taking into consideration 3-dimensional structure of protein or the target, conformational flexibility of the ligand molecule, presence of water molecules, presence of ions, and so on. We analyze the cavity in a proper way, try to place the ligand molecule, try to move the ligand molecule, compute its free energy, and then come with the right compound. Actually, dihydrofolate reductase like DHFR reductase inhibitors were designed by using such methods, and they are used in the disease malarial treatment and some other disease treatment rather. So, docking is basically we take a ligand, move the ligand into different points, calculate scores at each point, find base protein, compare scores, and different scoring methods are used, different optimization procedures are used, and the candidates are found. One of the hep word these days is pharmacogenomics. What is pharmacogenomics? In an experiment, in a DNA, we use a microchip, and gene sequence related to a particular disease is identified. Then this particular sequence is taken, it is converted into amino acid sequence. From the amino acid sequence, we try to find out whether it resembles to any existing proteins, do we have any structural information available about it. If we have the structural information, we take the information and then use drug designing method. In case we do not have any structural information, then we can use computer modeling technique to build the molecule in the active cavity of this protein rather. Now, we can try many such compounds and try to theoretically compute the activity and then come out with a drug which is a very efficient drug. So, pharmacogenomics is one of the most important method and important field of the present time. We can design drugs for the new genes which were not explored so far. We can design drugs for a specific patient and so on. So, personalized medicine can be developed and new drugs can be developed, new drug targets can be developed, and this is the only way by which we can enjoy the fruits of Human Genome Project. This was one of the biggest project of the last century, and this Human Genome Project was aimed at giving better service to the mankind, and if we want to put the information obtained to this project into the for the human mankind, we should go to pharmacogenomics.